Well, hello to everyone joining us and welcome to today's webinar. Our presentation is entitled Optimizing Rare Disease Trials, Trial Design and Execution. My name is Eric Falvey and it's my pleasure to be your moderator for today's session. Our event will run for approximately 45 minutes, including time for a Q&A session with our speakers. The session is designed to be interactive and works best when you're involved. So please feel free to submit questions for our speakers by using the Q&A panel as seen here, and we'll attend to your questions during the Q&A session. At this time, all participants are in listen-only mode. And a final note, please note the session will be recorded and made available to you for future viewing. So this session is proudly hosted by IDDI and August Research. So IDDI, the International Drug Development Institute, is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, specifically in oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, 30 years ago, IDDI is headquartered in Brussels, Belgium, with the US operations in Raleigh, North Carolina. And August Research, a boutique CRO focused on clinical operations in Europe and global pharmacovigilance, working across therapeutic areas, mostly in phase 2 to 3 studies. They offer a solid management team working together for over 15 years with very low staff turnover, and is a certified woman-owned business. So those are the two companies hosting this today. And now with this, it's my absolute pleasure to introduce you to our first speaker, Michael de Backer. Michael has a PhD in biostatistics and is a senior research biostatistician at IDDI. His research revolves around the statistical methodology called Generalized Pairwise Comparisons, or GPC, that he'll be presenting today, along with a case study on a particular rare disease. So Michael, I'll hand it over to you.
All right. Thank you very much, Eric, and thank you very much to all the attendees of today's webinar. It's my privilege to be here today to talk about a methodology that we've been developing and studying for a few years now with colleagues and collaborators. So the objective of my presentation today is twofold. First, I want to give you a glimpse of what GPC, Generalized Pairwise Comparisons, is all about. And then I'm going to show how this can impact the design of a clinical trial for a rare disease as an illustration of the methodology. So with no surprise, the talk will be divided in two parts. First, I'm going to introduce the statistical methodology, and then I'm going to apply it to a particular rare disease that I'll mention later. Now, for the first part, my objective is absolutely not to give as many statistical details as possible. I just want to give you an idea of what the methodology is all about. And the reason I want to do that is because it's a unique feature, a quite unique feature of the methodology, that it is able to be used as a tool for communication with non-statisticians, contrary to many complex statistical methodologies. This one, we can start discussing about it, and we can then also involve non-statisticians in all the process, which is quite, as I said, a unique feature that we really enjoy about the methodology. So to introduce the methodology, well, let me take you back to almost 80 years ago with the question of a two-sample randomized clinical trial. How can I show that my treatment is working better than my control? Well, a first way to reply to this question was proposed by WILCOXON, as I said, almost 80 years ago, with an idea that is quite simple, but is perhaps the most technical idea that we'll need to introduce in this talk. He said, well, if you're interested in having larger values for what you're going to measure, for instance, you're going to measure survival times and you're interested in patients surviving longer, then what you can do is just take all the values of your combined sample of treatment and control and order them. So you order all the values, and then what you will do is for each patient attribute a value that is equivalent to its position in this ordering. So for instance, if a patient gets a value of 1 called a rank, then that means that his survival time is the smallest survival time amongst all patients that you have studied in your trial. If he gets a value of 2, that means that he's the second smallest value, and so on and so on. And what WILCOXON said is, well, just look at the positions of all the patients under treatment and then decide if these positions are large enough for you to decide that this is not the play of chance that is at stake. It's the effect of your treatment that makes these patients survive longer. So in mathematical notations, WILCOXON said, consider the ranks of all the patients under treatment, add them up, and if this value eventually is large enough, then you can conclude that this is not the play of chance. Now, this is the first methodology, but it's quite complex. In some sense, you need to introduce the notion of ordering, ranking, and so on and so on. And that's not the easiest way to communicate with non-statisticians. Luckily, a couple of years later, MANN-WHITNEY, they showed that this methodology is completely equivalent to something much simpler to apprehend at first, is the idea of doing pairwise comparison. So what they say is, if you want to study if your treatment leads to longer survival than your control, then just take one patient from the treatment group, one patient from the control group, and compare the survival times of these two patients. And then once you have formed this pair, take a decision and attribute a value around this decision. If the survival time for the patient under treatment is longer than the survival time for the patient under control, then attribute to the comparison a value of 1. If it's the other way around, then attribute a value of 0 to the comparison. And if there's no difference between the two patients, attribute a value of 1/2. You do that exercise of constituting a pair and then comparing the values inside a pair. And you do that exercise for all possible pairs of patients you can form between one in the treatment group and one in the control group. So there's an idea that's already that's already easier to apprehend. It's basically you form pairs and then you decide based on the information in the pair, which of the two patients is the one better off. And if the treatment patient is doing better, you attribute a value of 1, 0 if it's reversed, and 1/2 if there's a status quo. Eventually, you aggregate all these values into one unique value that is the sum of all comparisons divided by the number of comparisons. And then you ask your statistician, once I have a number here for all this aggregation, is this large enough for me to conclude that this is very unlikely to be just the play of chance that's going on here, and I can then conclude that indeed my treatment is working better than my control. So that's the core idea, is forming pairs and then comparing them. And then we are now ready to start generalizing. So generalized pairwise comparisons, our starting point is pairwise comparisons, and we are going to generalize it. And I'm first going to generalize it in a very simple way that's not going to be very spectacular, but just for easier communication. What I'm going to do is just change this rule here of 1, 0, and 1/2, and I'm going to bring that on a scale that's easier, because in this rule particularly, 1/2 represents the status quo. And for me, it's easier to say, well, you know what, status quo, let's put it at 0. Let's put a perfect treatment at 1 and worst ever treatment at -1. So I'm just changing the scale, but I'm doing the same exercise. I'm forming pairs, I'm comparing, and so on. And then I'm just attributing a value no longer 1, 1/2, or 0, but just 1, -1, or 0. That brings me to the fact that 0 is my status quo. If it's positive, treatment is doing better. If it's negative, control is doing better. So that's not a very impressive generalization. What's more important is that in a second way of generalizing, what I'm doing is I'm changing from mathematical notations to text. So instead of writing xy greater than yj, I'm just replacing here by a text a notion of xy is doing better than yj. It seems like not much of a big step that I'm taking, but in fact, now I can start thinking about what it means for me to be doing better. Be doing better can be very arbitrary. I don't have to be, for instance, just larger on one endpoint on survival. I can start defining the notion of being better on a very different, on a very arbitrary scale that is relative to my study. Let me take you through the examples of how I can define the notion of better off. Well, for starter, I can, if I'm still studying just one endpoint, I can start taking the fact that, well, I don't need my endpoint to be just of one type. I can start playing around with endpoints of different types, that they can just be binary, they can be time to event, and so on. I don't need to have a restriction to one type of endpoint, as long as it is for me possible to define what it means to be better off on the scale of the endpoint. I can use my rule. So that's the first generalization. Though it's again, that's the most spectacular one, but then comes a second generalization that's already more important. And it comes from the idea of saying, well, if you were to give me the information of two patients, for instance, their survival times, and you were to ask me which one is doing better than the other, well, perhaps I will bring a nuance to my decision. I will say, well, you know what, if the survival times that I observe are just different from 1 week, is that truly a difference that matter to me? Well, depending on the type of disease, 1 week is perhaps not something spectacular, not clinically relevant. So what I can do is, when I bring this notion of being better, I can start nuancing. I can start saying, well, I don't need to be just better in survival times than the other patient. I need to be better by a certain amount. So I can bring to the picture what we call a threshold of clinical relevance that we denote here by this delta. And that's just the idea of saying, well, there needs to be a difference of at least a certain amount for it to be clinically relevant, for me to start saying, well, this patient is better off than the other one. For instance, the difference in survival times needs to be at least 1 week, 1 month, 6 months, depending on the context of the disease. But the rest of the procedure is exactly the same. I always do pairwise comparisons. I always attribute a value 1, -1, or 0, and I always aggregate them and ask my statisticians at the end if the number I get is large enough not to be due to the play of chance. So that's the second way of generalizing, is to say, well, better off means for me, well, better off by at least a certain amount, depending on the context of what I'm measuring. And now the last generalization, and that's the most interesting one, is again starting from the point that if you were to ask me which patient is doing better when you were giving me the information about 2 patients, it's very unlikely that my decision will be just on survival. For instance, if I measure toxicities, I will of course take them into account if survival times are quite similar. So if my 2 patients that I observe have survival times that only differ by 1 week, what I will do when you ask me to decide which one is better off, well, I will just look at toxicities, because survival times are similar enough for me to allow myself to look at toxicities. And what I'm doing there, what I bring fundamentally to the picture, is the fact that I'm no longer restricting myself to one unique endpoint when I define the notion of which one is better off. I bring several endpoints and I'm bringing them in an ordered way, because survival for me is the most important endpoint. If I don't have a survival gain that's big enough according to my context and my preference, then I allow myself to look at a second endpoint. That's exactly what we present here in this table when we classify pairs according to, by lack of better terminology, what we call win or losses, these values 1 or -1. Well, if the survival times are different enough, then I will just classify the pair based on that endpoint. If the difference in survival times is not large enough for me to make a decision on just this endpoint of survival, then I allow myself to look at the second priority, the toxicity endpoint, and then I will classify the pair as either a win or a loss and attribute a value of either 1 or -1. If I cannot do that, I attribute a value of 0. So when I think about this pairwise comparison exercise, it's very conceptually, it's very simple. You just take 2 patients and you decide which one is better off, and you attribute a value of 1 if the patient under treatment is better off, -1 if the patient under control is better off, and 0 otherwise. And this decision of which one is better off, this can be tailored really to the context, and in many cases an ordered definition of endpoints makes sense, because it really translates our thinking when we compare values of 2 patients. Eventually, we summarize all these pairwise comparisons, and what we like particularly as a summary measure is what we call the net treatment benefit. The net treatment benefit can just be seen as an average of all these pairwise comparisons, or in other words, the number of wins I will get minus the number of losses I will get divided by the number of pairs. So in the most profitable situation for the treatment, this value will be equal to 1. In the less profitable situation, this value will be equal to -1. And again, status quo is at 0. Now, for the sake of being complete, the idea of GPC has also been translated in other statistics. We use the net treatment benefits. If you're familiar with some literature, then you may have encountered the notion of win ratio. It's exactly the same idea of doing generalized pairwise comparison, but the summary measure is different. It's just the number of wins divided by the number of losses instead of the net treatment benefit. So that means that you have a relative effect instead of an absolute effect. Another notion that has been introduced as well in literature is the win odds that is tightly related to the net treatment benefit. So just to be complete about that in case you encounter them. And if you are one of the statisticians to which people will ask when is my summary measure big enough not to be due to the play of chance, then you can look at the literature about inferential procedures that we have listed here. Okay, so that's the first part of the presentation where I wanted to give you a glimpse of what GPC is all about. It's constituting pairs and defining a rule that will decide if pairs are in favor of the treatment or in favor of the control, or giving you to a status quo. Now, how can this impact the community of rare disease? Well, let me take an example of a particular rare disease that's called the SANFILIPPO syndrome. So in a nutshell, the SANFILIPPO syndrome is a rare disease that affects children of a young age. And schematically, what happens is that it affects in particular the development of children. So if you think about the development of a child, well, throughout the first years, there's an acquisition of different skills. For a child, schematically, acquisitions add up. For children that are affected by the SANFILIPPO syndrome, at some points, well, the development starts to slow down, and eventually there's a decline in the capabilities, in the acquisitions of children. Eventually, children affected by the disease usually live no longer than in their teens. So it's a very severe disease, and it's a very complex one because you're trying to measure a development. Development, of course, can be measured on many different scales depending on what's important for a notion of development. So we can see that this falls right into the target of generalized pairwise comparisons, where what we do is combine different endpoints of interest. Now, what is usually done to assess if a treatment is preferable than a standard of care for this type of disease is a summary measure called the development quotient. So it's really an aggregation of different scales that lacks a lot of granularity, because it's just an aggregation of different different scales, and you have no idea of what's playing around when you have a value for the development quotient. You have no way to decompose what happens into this development quotient on the different scales you are actually measuring. So what we did for this project was first to sit around the table with the medical people involved in this syndrome and define what's important to patients, and in particular their parents in this case, because we're talking about children. And a consensus was to say that, well, 5 domains are really of interest when we talk about the development of children. The first domain is the expressive language, so one's ability to express oneself. And that's really an important domain for parents of these children, because they of course need to understand what their child is going through. So the ability for children to be able to express themselves is really a key component of the disease. Other domains of interest include the motor skills, daily living skills, sleep, and pain. And these are all domains that are measured on very different scales, so very different components. But when you think about, well, generalized pairwise comparisons, in fact, you're bringing all these components on a similar scale. So that's an argument of using general pairwise comparisons, in addition to, well, ensuring that what you will show at the end can be decomposed into the effects of each of the domains that you're considering. So once we have done this job of defining what's important for the medical people involved in the syndrome, well, we need to do our statistician's job, and we need to have assumptions of what's going to happen in the control group, assumptions of the treatment effect we are supposing is going to happen, and then define what kind of sample size we will need to be able to detect that treatment effect with a certain guarantee, a certain power. So we need to start from somewhere, and luckily for this project, we had historical data. So that means that we had an idea of what a control group could look like. And then we needed, with, again, the medical people, we needed to define potential treatment effects that could happen within novel therapy. Now, the data is quite complex, so I'm not showing the data for the sake of time, but the data is quite complex because we're talking about development, and in development, there's a notion of time. That means that what we have eventually is data measured at different time points, and that makes the data quite complex, and complexity also arises on the fact that we have different domains, and these domains are interrelated. If a child has a strong deterioration in one domain, there's a high chance that he will also have a strong deterioration on another domain. So data is quite complex, and we need to account for that. Here, what I'm showing is what we got as information about what is expected for the treatment effect on each of the domains, and what would this result as a sample size for each of these domains. So the medical people were quite optimistic about having an effect on language, because the sample size required for a design that would only include language would be quite moderate. For other endpoints, for other domains, the assumptions are more moderate. Now, of course, we don't want to make a design just on language, because we don't want to replace the development quotient just by one of the domains. We want to replace the development quotient by something that makes more sense, that has more granularity than this development quotient, where everything is aggregated with no notion of what's playing around and what's driving the development quotient into positive or negative values. That's a reason why we propose to use generalized pairwise composite. As I said, the arguments for using a GPC, well, we have a single treatment effect that's interpretable. Of course, here, by lack of time, I'm going over some details that you may miss in order to have the full granularity of what's at stake. But still, you have the idea of what we do is doing pairwise comparisons on different domains. We order domains by importance. So we're told, for instance, language is the most important one. That means that we're going to put this as the top in our hierarchy of endpoints for our GPC decompositions. And as I said, another argument for using GPC is that initially these domains are on very different scales, while with GPC we bring that on a unique scale, and this is again quite a unique feature of the methodology. Eventually, when we make these assumptions and when we look at the sample size that would be required for the design of the trial, we end up with something that's very moderate in terms of numbers, and which is, of course, good news given the rarity of the disease. We ended up with a result that's saying that with 17 children for each group, we would have a power of at least 80% to detect the treatment effect that was assumed. Now, one last detail before concluding. You may ask, why do we aggregate all these endpoints in an ordering, and why do we not consider them separately, all these domains? Well, the answer lies in the fact that, again, there's a strong dependency between the domains. Children that are doing badly on one domain are very likely to be doing badly on the other domain, and that's represented here schematically by the fact that pairs that are considered tie, for instance, on the language domain, which is the most important, will be decomposed on the motor skills, and correlations enter into account in the fact that, well, these domains here are highly correlated with each other. So just to give you a glimpse of what happens if you consider these domains not to be interrelated. So if you do the same exercise of designing a trial and you ignore the correlation, then you have a sample size that is really unrealistic, because you will end up with a required sample size much larger than what we obtain when we truly account for the correlations between the domains that compose the net treatment benefits. All right, so to conclude this very short presentation on GPC, so GPC is all about forming pairs and then comparing pairs and classifying pairs, and that's something that's quite easy to communicate if you take the time, and it also brings the opinion of other stakeholders than statisticians into the picture, because the notion of doing better off than another patient, that's very arbitrary, and that can be tailored to every scenario that we encounter in practice. So that increases a lot of flexibility it incorporated multiple outcomes, as we've seen in the case study. An interesting aspect is the introduction of thresholds of clinical relevance, because not every difference, numerical difference, is clinically meaningful. And when you aggregate multiple outcomes, of course, the idea is that you incorporate more information into your analysis, and more information means less variability, which in turn means that you have potential to increase power as compared to using just a single endpoint. Eventually, this increase in power is also accompanied with a meaningful treatment effect, which is very desirable. And the interactions we've had so far about GPC, as I said, are quite favorable with both the FDA and EMA. Perhaps we can come back to that in the Q&A. So with that, let me thank you all for your attention, and I'll answer gladly any question you would have. And I'm giving you the floor back, Eric. Thank you.
Thank you, Mikhail. That was great. And so now it's my pleasure to introduce our second speaker, Angelina Lambine. Angelina has been is a senior project manager at August Research since 2012. She previously worked at PPD and ABCRO as a CRA. And prior to her 16 years in the CRO industry, Angelina was an ICU nurse at one of the biggest hospitals in Bulgaria. Angelina has a BS in nursing and an MS in clinical trial management and healthcare management. She's managed an extensive amount of rare disease studies and has seen the clinical trials industry from many perspectives. So Angelina, the floor is yours.
Thank you, Eric. Okay, so now we are going to talk about the execution of rare disease trials in Europe. And over the next 20 minutes, we are going to cover country selection, investigator identification, a little bit about the European regulatory process now, and some notes about trial execution and some key takeaways. So the first thing when you want to start a rare disease trial is to think about which countries in Europe you want to run your study. So a good starting point would be to have a look at the standard of care in the target countries and what treatments are available. You have to ensure that your protocol is feasible, that if there is specific genetic testing, which in a lot of rare disease studies, that is the case, that this genetic testing can be done in these particular countries, or if there are specific exams that this can be done. So these are the points that it's good for you to consider when selecting the countries, as these really vary within Europe and there are differences. Also, you may find the suitable patient population in some countries where you can't run your trial, and you might think of the possibility to send patients from that country to another country, neighboring country, on another country where the study will be running. We had such case on a rare disease pediatric trial where we had potential children in Bulgaria, and we sent these to Turkey where the study was running, and we used some concierge services for that. And in such cases, you also have to make sure that you have all the documentation, and in particular the consenting documentation, because of the languages, which in Europe, in most of the countries, the languages are different. Another good point to consider is the use of home care nursing services, because we see that a lot in the rare disease trials, especially where there is patient population who have disabilities, who cannot easily go to the clinic for the study visits. And these services really help a lot, but you have to make sure that these home care nursing services are allowed in the particular countries, because in Europe, there are countries like Bulgaria where such services are not allowed. Another good point to consider when selecting the countries is to review the formulary, and we've seen clients focusing countries which do not have the cutting-edge pharmaceutical approved or available at the country. And in this situation, your trial really may become really attractive to the investigators. Also, you may want to consider Central and Eastern Europe, where fewer expensive treatments are available. And in such situations, your rare disease trial may also be very attractive to patients and to investigators. Okay, so once you have identified the countries where you want to run your rare disease trial, then the next step is to do some feasibility. So we've seen a lot of sponsors send blast emails with surveys, and according to us, we don't think that's really sufficient for a number of reasons. If you're a small sponsor and your name is not well known, your questionnaire may get ignored very easily because the investigators are busy. They get a lot of emails, a lot of questionnaires, and your questionnaire might not be answered that easily. So what do we think, and what we encourage sponsors to do is to have that personal approach, like what we are doing. Calling the sites and presenting the protocol, presenting the design, and talking about the suitable patient population. And in that cases, it really helps to get your questionnaire very quickly answered. So what we can say that it's very important to check if the CRO that you're going to work with has those personal relationships with the sites. Another important thing to consider when doing your feasibility is to check if sites can pre-screen or pre-select patients. And in such situations, like we had on some of our studies, sites, they already had pre-selected patients, and then really led to shorten the enrollment period. So this is also something that you can consider when preparing your questionnaire. And the last thing is sometimes you can contact sites which have the patient population, but they really don't have the resources or the ability to participate in that trial, and they can refuse. But you can talk to them, and if they agree to refer patients to other sites within the country. We had such case in UK on a rare disease study where we did that, and during this feasibility phase, we agreed with some sites which had the potential patients to actually refer patients to other sites in UK. Okay, so you have identified your countries, you've been through your feasibility. Then the next piece is to do the regulatory steps and submit your study and get your review and your approvals in place. So as some of you from Europe may know, the regulation that was in place since 2009 and is still actually in effect, it allows for sponsors to submit the study in separate countries at different time, to do your ethics committee submission, to do your regulatory submission, and to prioritize countries where you can get your approvals quickly. So, but now having the new clinical trial regulation, which came into force since 2022 and will become mandatory as of February 2023, it requires a centralized submission through a single portal for all European countries. So what's specific is that you have to collect all of the documentation for all of the countries that you're considering to run your study and to do your submission at once. Meaning that, for example, if you want to run your study in France and Germany, then you have to collect all of the documentation, you have to prepare everything and to do one single submission at the same time. So that has some negative part that you really cannot there is no chance for faster startup from the quick approving countries like what we have been doing so far. We can still do, but as of February, as I said, February 2023, this will not be possible, and one single submission will be mandatory. And if you want to add other countries, then you have to wait for your initial approval, and only after that to do this additional submission. Also, it's important to mention that there is one new thing, and this is that sponsor must submit 2 versions of the study documents. One version, one package will be for the drug agency's review, and the other version of the documentation will be for the public consumption. So that increases the transparency. This is something new, which all sponsors will be mandatory to comply with starting from February 2023. And this is good to mention that this will cost more work than before, but this will be the situation as of next year, as I said. So the new clinical trial regulation in Europe works with a system called Clinical Trial Information System, or CITIS. And this is the single portal where you have to submit your study. And for the people who work in Europe and know that previously we were preparing packages for one package for ethics committee, one package for the regulatory agencies. Now these documents are the same, they're just called differently. They're called Part 1 package for the drug agencies and Part 2 for the ethics committees. And as I said during the previous slide, that this submission will have to be done at once. Okay, and here on this slide, you can see the timelines for your clinical trial review and approval. If it is a regular trial with a regular medication, then you can see that the timelines are from 60 to 106 days. And if it is with an advanced therapy, then the timelines for review and approval are from 60 to 156 days. What's important to mention is with regards to the contracts, site contracts, this will still have to be done in parallel while waiting for your review and approval. And a good point to consider is that, especially on rare disease studies, when you work with huge university hospitals, they really have a very slow turnaround time for contracts negotiation. So it's good for you to consider that, and especially in some countries where we see very slow period for that, from 3 to 6 months. So we just recommend that you have this in mind. Okay, and I would like also to mention that the new clinical trial regulation, it contains specific wording with regards to the rare disease trials. And we recommend that you have a look at it when you're designing your study, preparing your protocol, so that you can ensure that documents are in alignment with that regulation and what's in there. And also would like to say that the European Union are really focused on fostering the development of orphan medicinal products for ultra-rare disease, and this is specified as 1 person in 50,000 in the European Union. And also to say that particular experts will be responsible for that review. So just to make sure that your trial will be reviewed accordingly. And I'm not going into other details. For more information, you can refer to the regulation, and you can see that following the link that you can see on this slide. And the last topic that I would like to touch base here with regards to the rare disease, this is the pediatric population and the consenting. And the pediatric population is very important for the rare disease. As you know, a lot of studies are done with pediatric populations because of the genetic origin. And there are specific requirements for the ICFs and for the ASCENT forms, which really can vary per country, and they vary according to the ages of the children. So they vary, as I said, within Europe. You have to make sure and check in the countries where you'll be running your study, that if you're meeting all the requirements. And also to mention that it's important to check specific requirements, like if both parents will need to give consent, or one parent will be allowed to do that, because we see that really challenging, especially in situations where, for example, one parent is working in a different country. And here on this slide, you can see the age ranges. They really differ, as I said, in each of the countries. So different assent forms will have to be prepared. So you have to look at these requirements and make sure your documents meet these requirements. And here, for example, in Spain, you can see that only one of the parents has to give consent. And in other countries, which are just examples here, like France and Netherlands, both parents have to give consent. Okay, so you've been through your country selection. You know which countries will be participating in your trial. You've done your feasibility. You've been through the regulatory process where you've got all your approvals in place. So now it starts to the study execution, to activate your sites and to start enrolling patients. So it's really important to mention that sponsors cannot delegate all of the responsibilities to the CRO. And you just cannot delegate everything to the CRO and expect at the end of the study that you'll get your reports, your data, and everything will meet your requirements with the big bow on that. That's not the case. You really have to be involved into what's going on, into your study, especially for rare disease studies. And especially in the cases, if you're a small sponsor and not very well known, then you really need to foster your relationships with the sites and to promote engagement with that, with the trial. So what we have been seeing, like it's working, and what we can encourage sponsors to do is, first, for example, use the pre-screening and screening clocks, especially in the cases if your study is long and if the enrollment is very slow, then the investigators, they may forget about your study easily, and without asking them to complete these forms, you remind them about your study. So this could be working. And we also encourage sponsors to call the sites, to organize direct calls with the sites. We arrange also such calls. We've seen that a lot, and we think that's working because the investigators, they really appreciate that attention and that personal approach, and they can share concerns, they can share what hurdles they're facing, and they really appreciate that, as I said. We've seen also a lot of sponsors organizing roundtables with investigators. If there are multiple sites within a country, they organize such roundtables. They meet with investigators personally, and the investigators, they appreciate that they can directly interact with the sponsor. They can share their experience if they have questions, if they have concerns. So that's really much appreciated, and this really can show your your engagement with the trial and with the sites. And the last thing is, we recommend sponsors to use clinical trial liaisons. And these are people who could be hired or could be from your own resources. These people could be with medical background or just to be trained for this particular protocol. And with them, you can really build your relationship with the sites on the ground, to promote your trial, to have that personal approach, and to interact directly with investigators, and investigators feel appreciated, and with this they can have their direct contact with the sponsor. So these are the few things that we encourage sponsors to do. We think these are working effectively for building your relationships with the sites and showing your involvement. Okay, so according to ICH GCP, you know that sponsors, they have the ultimate responsibility for conducting the clinical trial. You cannot delegate that ultimate responsibility to a CRO. So you really have to maintain your oversight during the study conduct. And you can do that with the regular meetings with the CRO or with your vendors. We always use written agendas and meeting minutes, but you really have to dig into details. You really have to make sure you understand what's going on and to review this information. Similarly, you receive study metrics and monitoring visit reports and review the deviation logs, all these outputs, but you have to deeply review this. For example, do some spot checks on the monitoring reports so that you really make sure you understand what's going on at the investigational sites and there is no difference between what the CRO is telling you. So this is very important for you to maintain your oversight. And another thing that you can consider in order to maintain your oversight role is to do some co-monitoring visits with the CRO study monitors. We also think that investigators appreciate that. It's again another kind of personal approach, and they appreciate and they feel important, and again they can share their experience, their hurdles. So it's another good point for you to consider. Also, another point that you can think about is to use the clinical trial liaisons, as I just said during my previous slide. That's really helpful to use these people, build your relationships on the ground, and to confirm your oversight. And I would like also to say that maintaining this oversight and having these responsibilities, it doesn't have to lay only with the clinical team. I mean, you can really share that with the quality people or with CMO. We've seen that a lot. So you have to understand that it doesn't have to, bring you additional burden and to lay only with the clinical team. Okay, so these are the things you can consider, using in order to maintain your oversight role during the conduct of the study. So before going to the questions part, let me just review some key takeaways from this presentation. So first, it's very important for you to consider in which countries in Europe you're going to run your rare disease trials. So this really can be, a successful key point for you to consider. So it's very, we recommend that you really check and see where in Europe you can run your study, which countries you can select. And the new clinical trial regulation in Europe, this is new system. It's new for the industry. We are all adjusting. It's a learning process, but it is expected to streamline the approvals. And the last thing is, as I just said, the close sponsor oversight. That's really important for you in order to have a successful enrollment and a successful study. So thank you very much for your attention. I really appreciate that. And now going to the questions part. Thank you again.
Great, thank you, Angelina and Mikhail, for your excellent talks. If we can, let's move over to the Q&A session here. Sorry, I'm a little computer issue. So let's begin the Q&A portion. Just to remind the audience, you can always send us questions using the Q&A panel there. And we've already got some questions, so let's start with those. Well, I guess this is top of mind for many people. How are the clinical trials in Europe affected by the war in Ukraine? So Angelina, that's probably one for you. I think you're on mute.
Yeah, yeah, sorry, I was on mute. Well, it's this situation cannot be ignored. And we can say that Ukraine and Russia, they were huge contributors of patients especially for the rare disease trials, but now they're off limits because of the situation. And these countries, together with Belarus, because of the sanctions, we think these are off limits and they can not run so many rare disease studies as they did in the past decade. And also the non-NATO countries like Georgia and Moldova, they could have a higher risk profile for conducting clinical trials. But we have an office in Georgia and things are running smoothly thus far, and we are hoping the situation stays the same. And what I can say is that the NATO countries, they're really a safe destination for conducting clinical trials.
Okay.
Thank you.
Thank you, Angelina. Here's one for you, Mikhail. There's several here. So is GPC applicable to more than 2 groups comparison? And how do we adjust for multiple comparisons?
It's an excellent question. In its current form, not yet applicable to more than 2 groups. And it's a complex question of how you extend that to more than 2 groups, but it's not one that is that we're not going to be able to tackle. The complexity is not per se the multiple comparison. The complexity is, in technical terms, the potential non-transitivity, meaning that treatment A can be declared better than B, B better than C, and C better than A. So there's an untransitive issue there, and that's something that's lingering around when you play with probabilities, and that's something that's that people have studied outside of the GPC framework in univariate settings, and that we can use to extend the GPC procedure. But in its current form, it's 2 samples, but there are the foundations to extend it. It's just a matter of research.
Okay, thank you, Mikhail. Here's a current one. Are you running trials in Moldova at the moment, and what is the situation there? Another one for Angelina, I imagine.
Yes, I can say that we are still running trials in Moldova. We haven't seen any changes in that. We have feasibilities. We are having an ongoing study, and we haven't seen any differences since this conflict started.
Okay, thank you. Here's one for you, Mikhail. Maybe you can talk to this one. Could you tell us about the regulatory's position regarding this novel methodology you promote?
Yeah, sure. So I mentioned it very quickly in the last line that we have had positive interactions with the EMA and FDA ourselves. So interactions with our group at IDDI. But there are also other research groups that are interested in GPC, and this is gaining much traction in other domains. For instance, in cardiovascular studies, a few studies are now accepted by the FDA where GPC is used as a primary endpoint. So studies are really conducted around GPC as we are speaking now. So there's a real traction that's very exciting and very interesting from the regulatory point of view.
Excellent. Okay. Angelina, here's one for you, I believe. How can CTLs improve recruitment?
Well, we have a lot of cases using such people, and they go and meet with investigators, and they don't talk about open queries or missing pages. They just talk about the study and the protocol and what hurdles they're facing. For example, we were involved as a company separately from the CRO managing the study. As a company, we are involved to only do the clinical trial liaison part, and I was personally involved in managing such a case where we helped first to resolve and to speed up the contract's execution, and then we were in direct contact with investigators and helping them answering questions very quickly with regards to patient eligibility and asking them to identify patients. You know, the other CRO was a really big CRO. They didn't have that quick interaction that we had with the sponsor. So that's a really a good point to consider. And in our cases, this really helped to boost the enrollment. Thank you.
Excellent. Thank you, Angelina. Here's one for you, Mikhail, and maybe we'll make this the last official question. we'll follow up after this with the other ones in an email, but how do you determine which one outcome is important over another when we have a tie in primary outcome, say survival?
Well, that really depends on what's important for you, and how you would decide implicitly if you were to be given the information about two patients and you were to be asked which one is the one that is better off. Then implicitly what you're going to do is some kind of hierarchical thinking that we can translate into mathematical terms, but that really depends on the context and what is important to you. And the notion of importance to you is quite unique, because that means that you can really tailor the analysis depending on what's important to you, which may be different from your neighbor. And so once study is approved and so on, you can also use it to really tailor decisions based on what is important to you, which may again be different from your neighbor. So that's a really specific preference that you need to define for yourself.
Okay, thank you, Mikhail. Maybe, Angelina, there's one more. Maybe we can close out with this one officially, and then we can get to wrap up here. Do you use patient advocacy groups to help support rare disease trials in Europe?
Yes, we do. We have experience with such groups in all of our countries, and we establish contact with them, and they can recommend to patients to participate in this particular trial. So yes, I can say we have relationships and we have experience with such advocacy groups. Thank you.
Okay, great. Well, I'm just conscious of the time here, so I think we need to stop here. And please note, we had several other questions come in, but I think we will definitely follow up with everyone following this webinar as a follow-up. But just wanted to say thank you again to Mikhail and Angelina for your wonderful presentations. And as a reminder to everyone, we'll have a webinar recorded, so this will be sent as a follow-up. You'll have access to the recorded archive of this event. We hope you found this webinar informative. It's been my absolute pleasure to be your moderator. So on behalf of IDDI and August Research, we thank you for joining us today. I'm Eric Falvey, and until next time, bye for now.